Hey there, night travellers. Before we begin tonight's journey into the most complex structure in the known universe, take a moment to like the video and subscribe if you genuinely enjoy these deep explorations into what makes us human. Drop a comment letting me know where you're watching from and what time it is for you. It's always fascinating to know who's joining us on these nocturnal voyages through knowledge. Now settle in, dim the lights if you can, maybe turn on a fan for that soft ambient hum. Tonight we're going somewhere extraordinary and we won't be taking any shortcuts. We're diving deep into the 3 pounds of tissue sitting inside your skull right now, processing these very words, converting photons into meaning, creating the experience of consciousness itself, the human brain. It's been called the most complicated object in the universe, and for once, that might not be hyperbole. 86 billion neurons, each one connected to thousands of others, creating a network so vast that the number of possible connections exceeds the number of atoms in the observable universe. This isn't just an organ, it's a cosmos contained within bone, and tonight, we're going to explore every major region, every crucial function, every mystery that science has managed to unravel so far. Let's begin where complexity reaches its zenith. Part 1. The Cerebrum, the crown of consciousness. The cerebrum sits atop your brain like a wrinkled crown, and it's massive. Accounting for roughly 85% of your brain's total weight, this structure is what makes you distinctly, recognizably human. It's where your personality lives, where your memories form constellations of experience, where every conscious thought sparkles into being before fading back into the neural darkness. When you look at a brain, that characteristic walnut-like appearance comes from the cerebrum. Those ridges and grooves aren't random wrinkles. They're Giri and Sulci, evolutionary adaptations that pack an enormous surface area into the confined space of your skull. If you could unfold the cerebral cortex and lay it flat, it would cover about 2.5 square feet, roughly the size of a large pizza. Nature needed to fit all that neural real estate into a space about the size of a small cantaloupe, so it folded it, over and over again, creating the convoluted surface we recognize as brain matter. The cerebrum contains approximately 16 billion neurons in its cortex alone. Each of these neurons can form thousands of synaptic connections with other neurons, creating a network of mind-boggling complexity. If you tried to count every synapse in the cerebral cortex, counting one per second, it would take you roughly 32 million years to finish. And that's just the cortex. The computational power represented by this neural network dwarfs any computer ever built or even conceived. But the cerebrum isn't a uniform structure. It shows remarkable regional specialization with different areas handling different functions, yet these areas don't work in isolation. They form intricate networks, circuits that span multiple regions and even multiple lobes. Understanding the cerebrum means understanding both the specialization of individual regions and the integration across the whole structure. The cerebrum divides itself down the middle into 2 hemispheres, left and right, separated by a deep groove called the longitudinal fissure. These hemispheres don't work in isolation. They're connected by the corpus callosum, a massive bundle of 200 million nerve fibers that allows the 2 sides of your brain to communicate. Think of it as the world's most sophisticated fiber optic cable, constantly shuttling information back and forth, allowing the left side of your brain to know what the right side is experiencing and vice versa. The corpus callosum isn't the only connection between hemispheres, though it's by far the largest. The anterior commissure, a smaller bundle of fibers, also connects the 2 sides, particularly the temporal lobes. The hippocampal commissure connects the 2 hippocampi. These commissural fibers are essential for integrating information from both sides of the brain, creating a unified experience of the world rather than 2 separate consciousnesses. We know this because of split brain patients, individuals who had their corpus callosum severed as a treatment for severe epilepsy. These patients, studied extensively by Roger Sperry and Michael Gazzaniga, revealed fascinating insights into how the hemispheres function. In everyday life, these split brain patients seem relatively normal, because sensory information reaches both hemispheres through the brainstem. But in carefully controlled experiments, researchers can present information to only one hemisphere, show a word to the left visual field, processed by the right hemisphere, and the patient can't verbally identify it because language is in the left hemisphere. but they can draw it or point to a matching object with their left hand controlled by the right hemisphere. It's as if two separate minds inhabit the same skull, each with access to different information and different capabilities. Here's something remarkable about these hemispheres. They exhibit what neuroscientists call contralateral control. The right hemisphere controls the left side of your body, and the left hemisphere controls the right side. When you move your left hand, that command originates in your right hemisphere. When your right eye processes visual information, much of that data gets sent to your left hemisphere. This crossing over happens in various places throughout the nervous system, and while we understand that it happens, the evolutionary reason why remains one of neuroscience's enduring mysteries. But the hemispheres aren't just mirror images of each other. They show lateralization, meaning certain functions tend to be concentrated more in one hemisphere than the other. Language processing, for most people, happens predominantly in the left hemisphere. Spatial processing and face recognition tend to lean right. But here's where popular neuroscience has led us astray. You've probably heard people described as left-brained or right-brained, implying that logical people use their left hemisphere more while creative people live in their right hemisphere. This is neuromythology at its finest. The reality is far more nuanced and far more interesting. Both hemispheres work together for virtually everything you do. Yes, language centers tend to cluster on the left, but understanding the emotional tone of speech, the prosody and rhythm, that's a right hemisphere specialty. Mathematical calculations might favor the left, but estimating quantities and understanding numerical relationships involves the right. The brain doesn't respect the clean boundaries we like to draw. It's messier, more integrated, more collaborative than any simple dichotomy can capture. The outer layer of the cerebrum, the cerebral cortex, is where the magic happens. This thin sheet of grey matter, about 2 to 4 millimeters thick, contains the cell bodies of billions of neurons. It's organized into 6 layers, each with its own characteristic cell types and connection patterns. These layers work in concert, creating circuits of stunning complexity. Layer 1, the molecular layer, contains relatively few neuronal cell bodies, but abundant dendrites and axons from other layers, creating a dense web of connections. Layer 2 and 3, the external granular and external pyramidal layers, contain smaller pyramidal neurons that project primarily to other cortical areas, forming cortico-cortical connections. These layers are crucial for communication between different regions of the cortex, allowing information to be shared and integrated across specialized areas. Layer 4, the internal granular layer, is the main recipient of sensory information arriving from the thalamus. This layer is particularly thick in primary sensory corticas, where the first cortical processing of sensory information occurs. In primary visual cortex, layer 4 shows remarkable substructure, with sublayers that process different aspects of visual information. Layer 5, the internal pyramidal layer, contains large pyramidal neurons whose long axons project to subcortical structures like the basal ganglia, thalamus, and brainstem, as well as down to the spinal cord. These are the output neurons that allow the cortex to influence movement and other functions. Layer 6, the multiform layer, projects primarily back to the thalamus, forming reciprocal connections that modulate the sensory information flowing into the cortex. This feedback allows the cortex to influence its own input, filtering and prioritizing sensory information based on current goals and expectations. This laminar organization isn't uniform across the entire cortex. Different regions show different patterns. Motor cortex has a thick layer 5, packed with the large pyramidal neurons needed to send commands to the spinal cord. Sensory cortices have thick layer 4 to receive abundant sensory input. Association cortices, involved in higher level processing, show more balanced layer development. These variations in layering, visible under a microscope, allow neuroscientists to map cortical areas and understand their functions. Beneath the grey matter lies white matter, and here's where we find the brain's communication infrastructure. White matter gets its colour from myelin, a fatty substance that wraps around axons like insulation around electrical wires. Myelin does more than just protect. It dramatically speeds up the transmission of signals, allowing information to travel down an axon at speeds up to 270 miles per hour. Without myelin, your nervous system would be sluggish, unreliable, incapable of the precise timing that makes coordinated movement and complex thought possible. Diseases that attack myelin, like multiple sclerosis, demonstrate just how crucial this insulation is. As myelin deteriorates, signals slow down or fail to transmit entirely, leading to a cascade of neurological problems. The cerebrum organizes itself into 4 major lobes, each named after the skull bone that sits above it. Let's start at the front with the frontal lobe, the largest of the 4 and arguably the most important for what we think of as human behavior. The frontal lobe extends from your forehead back to a deep groove called the central sulcus. It's enormous, taking up about 1/3 of the cerebral cortex, and it's the last part of the brain to fully mature, not reaching complete development until somewhere in your mid to late 20s. This delayed maturation explains a lot about adolescent behavior, but we'll get to that. The frontal lobe underwent dramatic expansion during human evolution. Compared to other primates, humans have disproportionately large frontal lobes, particularly the prefrontal regions. This expansion is thought to underlie many uniquely human capabilities: complex planning, abstract reasoning, moral judgment, impulse control, and the ability to consider multiple perspectives simultaneously. The frontal lobe, in many ways, is what makes us human. At the very back of the frontal lobe, right up against the central sulcus, sits the primary motor cortex. This narrow strip of tissue is your brain's movement command center. It's organized somatotopically, meaning each part of your body maps to a specific location. Move your fingers, and a cluster of neurons in one section of the motor cortex fires. Wiggle your toes, and a different cluster activates. If you could see this map, it would look bizarre. Your hands and face would be enormous, taking up disproportionate amounts of cortical real estate, while your torso and legs would be tiny by comparison. This distorted representation, called the motor homunculus, reflects the precision with which different body parts need to be controlled. Your fingers require fine motor control for tasks like threading needles or playing piano, so they get more cortical space. Your back doesn't need that level of precision, so it gets less. The motor cortex doesn't work alone. It receives input from premotor areas, from the supplementary motor area, from the posterior parietal cortex providing information about spatial location, and from the cerebellum and basal ganglia providing coordination and action selection. The actual motor commands sent down to the spinal cord are the product of extensive processing and integration across multiple regions. Interestingly, the motor cortex isn't just active during movement. It also activates when you observe someone else moving, when you imagine moving, even when you think about movement in abstract terms. Mirror neurons, discovered in premotor cortex, fire both when you perform an action and when you watch someone else perform the same action. These neurons might be crucial for understanding others' actions, for learning through imitation, possibly even for empathy and social cognition. Just in front of the motor cortex lie the premotor areas, regions involved in planning and coordinating complex sequences of movements. When you learn a new physical skill, whether that's a tennis serve or a guitar riff, these regions are intensely active, working out the timing and sequence of muscle contractions needed. With practice, these sequences become more automatic, requiring less conscious attention as the patterns become encoded in the connections between neurons. The supplementary motor area, on the medial surface of the frontal lobe, is particularly important for internally generated movements, actions initiated by your own intentions rather than in response to external cues. It's active when you decide to move, when you plan a sequence of movements, when you coordinate movements of both hands together. Damage to this area can produce strange symptoms, where movements become difficult to initiate voluntarily, though reflexive movements in response to stimuli remain intact. But the frontal lobe's true magic happens in its most anterior region, the prefrontal cortex. This is where your brain does its most sophisticated thinking. The prefrontal cortex handles executive functions, planning for the future, weighing consequences, inhibiting inappropriate responses, maintaining focus on tasks despite distractions, adapting strategies when circumstances change. It's your brain's CEO. Though like many CEOs, it doesn't always make the best decisions, especially when it's tired or stressed or still developing. The prefrontal cortex shows remarkable specialization within itself. The dorsolateral prefrontal cortex, on the upper and outer surface, is crucial for working memory, the ability to hold information in mind and manipulate it. When you're doing mental, arithmetic, holding a phone number in memory, or following multi-step directions, this region is active. Patients with damage here struggle with tasks requiring mental manipulation of information. They can perceive and remember normally, but they can't maintain and work with information over short periods. Working memory isn't a single process, but involves multiple components. The dorsolateral prefrontal cortex maintains information in an active state through sustained neural firing. Even after a stimulus disappears, neurons continue firing, maintaining a representation of that stimulus. This persistent activity allows information to be held online and used to guide behavior. The capacity of working memory is limited, which is why you can only hold a handful of items in mind at once. This limitation appears to reflect the number of distinct neural populations that can maintain stable patterns of activity simultaneously without interfering with each other. The ventromedial prefrontal cortex, on the lower and inner surface, helps with decision-making and emotional regulation, integrating emotional information with rational thought. This region receives input from the amygdala and other limbic structures, allowing emotional information to influence decisions. Damage here can lead to poor judgment and difficulty learning from mistakes, despite intact intelligence and reasoning ability. The famous case of Phineas Gage, whom we mentioned earlier, involved damage to this ventromedial region. But the story of Gage is more complex and more interesting than the simple tale often told. Contemporary accounts suggest Gage's changes, while real, were perhaps not as dramatic as later retellings suggested. He did work as a coach driver after his accident, requiring planning, responsibility and social interaction. But he clearly wasn't the same person, and his case launched modern neuroscience's investigation into the frontal lobe's role in personality and social behavior. One famous case illustrates the prefrontal cortex's importance beautifully, though tragically. Phineas Gage, a railroad worker in 1848, had an iron rod blown through his skull in a blasting accident. The rod entered below his left cheekbone and exited through the top of his skull, destroying much of his left frontal lobe. Miraculously, Gage survived. Even more miraculously, he could walk and talk and reason, but his personality transformed completely. Before the accident, he was responsible, hard-working, well-liked. Afterward, he became impulsive, irreverent, unable to stick with plans or control his behavior. His intellect remained, but the person he had been was gone, replaced by someone his friends said was no longer Gage. The case demonstrated that personality and character aren't abstract properties of the soul, but concrete functions of brain tissue, particularly frontal lobe tissue. The orbitofrontal cortex, sitting just above the orbits of the eyes, is involved in processing, rewards and punishments, in learning from outcomes, in adjusting behavior based on changing circumstances. This region helps you learn which choices lead to good outcomes and which to bad, gradually shaping behavior through experience. The orbitofrontal cortex is particularly important for reversal learning, the ability to adapt when previously rewarded options stop being rewarding. If you learn that pressing button A gives you points, but then the contingencies reverse so button B gives points instead, your orbitofrontal cortex is what allows you to update your strategy. Patients with orbitofrontal damage often make poor decisions, particularly in social and personal contexts. They might say inappropriate things, make risky choices, fail to learn from negative consequences. In laboratory tests, they show impairments on tasks like the Iowa gambling task, where healthy participants learn to avoid risky decks of cards that occasionally yield big losses in favour of safer decks with consistent small gains. Orbitofrontal patients keep choosing the risky decks, unable to integrate the pattern of losses into their decision-making. The delayed maturation of the prefrontal cortex has profound implications for adolescent behaviour. During adolescence, the limbic system, particularly the nucleus accumbens and amygdala, reaches adult-like levels of activity. The prefrontal cortex, however, continues developing, with synaptic pruning and myelination extending into the 20s. This mismatch creates a period where motivation and emotional reactivity are high, but top-down control is still developing. This helps explain the increased risk-taking, sensation-seeking and emotional volatility characteristic of adolescence. It's not that teenagers can't reason about risks or understand consequences. In laboratory settings, teenagers reason about risks as well as adults do. But in real-world situations, especially emotionally charged or social situations, the still developing prefrontal cortex struggles to exert control over limbic drives and impulses. The cognitive brakes exist but aren't as reliably engaged as they'll become in adulthood. This developmental trajectory isn't a design flaw, but may serve important functions. The increased exploration and risk-taking during adolescence facilitate independence, learning, and the discovery of new opportunities. The heightened sensitivity to social information helps navigate the complex social landscapes of adolescence. Evolution may have shaped this developmental pattern to promote behaviors that, while sometimes risky, are important for the transition to adulthood. The frontal lobe also contains Broca's area, typically in the left hemisphere's inferior frontal gyrus. This region is critical for speech production, for turning thoughts into the motor commands needed to move your tongue, lips, and vocal cords in the precise patterns that produce words. Damage to Broca's area results in expressive aphasia, a condition where someone can understand language perfectly but struggles to produce it, their speech halting and effortful, reduced to simple phrases and individual words. Behind the frontal lobe, across the central sulcus, lies the parietal lobe. If the frontal lobe is about action, the parietal lobe is about sensation and spatial awareness. The parietal lobe's front edge contains the primary somatosensory cortex, a strip of tissue that mirrors the motor cortex's organization. This is where your brain processes touch, pressure, pain, temperature, and proprioception, the sense of where your body parts are in space. Like the motor cortex, it's organized somatotopically, with a sensory homunculus that gives exaggerated space to your hands, lips, and face, areas rich in sensory receptors and requiring fine discrimination. The somatosensory cortex doesn't just passively receive sensory information, it actively processes and interprets it. Different regions of the somatosensory cortex extract different features from sensory input. Some neurons respond to light touch, others to pressure, still others to texture or vibration. These feature detectors allow your brain to construct a rich, detailed representation of tactile experience. When you run your fingers over a surface, you don't just feel generic touch, but can distinguish smooth from rough, hard from soft, warm from cool. The resolution of somatosensory mapping varies dramatically across the body. Your fingertips can distinguish 2 points, separated by just 2 or 3 millimeters, as distinct touches. On your back, 2 points need to be several centimeters apart before you perceive them as separate. This reflects the density of sensory receptors in the skin and the amount of cortical territory devoted to processing information from each body region. The variation in tactile acuity makes evolutionary sense. Your hands need exquisite sensitivity to manipulate objects, your back doesn't. But the parietal lobe does more than just receive raw sensory data. It integrates that information, creating a coherent understanding of your body in space. The posterior parietal cortex toward the back of the lobe combines sensory input with visual information to construct your spatial awareness. This region lets you reach for objects without looking at your hand, lets you navigate through crowded spaces without bumping into things, lets you judge distances and sizes and relationships between objects in three-dimensional space. The posterior parietal cortex contains neurons with remarkable properties. Some neurons fire only when objects appear in specific locations relative to your body, creating a representation of nearby space. Others respond to objects relevant to your current goals, highlighting potentially important items. Still others combine information about where objects are with information about planned movements, helping to transform visual information about object locations into motor commands for reaching. This transformation from sensory information to motor plans is one of the parietal lobe's crucial functions. When you see a coffee cup and decide to pick it up, your parietal cortex processes the cup's location, size, and orientation, then helps generate the appropriate reaching and grasping movements. This sensoromotor transformation happens largely outside conscious awareness, but is essential for skilled interaction with the physical world. The parietal lobe also plays a critical role in attention, particularly spatial attention. When you focus your attention on one location in space, even without moving your eyes there, your parietal cortex shows enhanced activity. This covert attention without overt orienting, allows you to monitor your surroundings and shift focus rapidly when something important happens. The parietal lobe appears to maintain a priority map of space, with locations that are behaviorally relevant showing enhanced activity, biasing processing throughout the visual system. Damage to the parietal lobe can produce some of the strangest neurological symptoms. Hemispatial neglect, typically following right parietal damage, causes people to ignore the left side of space entirely. They might eat only from the right side of their plate, dress only the right side of their body, draw a clock with all 12 numbers crammed into the right semicircle. The left side doesn't cease to exist exactly. It simply fails to register as worthy of attention. What makes neglect particularly puzzling is that the basic sensory systems often work fine. Show a light in the neglected field and the visual system registers it. The eyes can see it, the neurons in visual cortex respond to it, but somehow the information doesn't reach awareness or guide behaviour. The deficit appears to be an attention in the system that determines what sensory information becomes conscious and influences action. Neglect patients often lack awareness of their deficit, a phenomenon called anosognosia. Ask them if anything is wrong, and they might say no. Everything is fine, even as half the world has vanished from their experience. This lack of awareness suggests that the parietal lobe isn't just processing sensory information, but is involved in self-monitoring, in maintaining awareness of our own mental states and capabilities. Another bizarre consequence of parietal damage is agnosia, specifically tactile agnosia. Someone with this condition can feel objects but can't identify them by touch alone. Hand them a key with their eyes closed, and they can describe its shape, its temperature, its texture, but they can't tell you it's a key. The raw sensory information arrives, but the synthesis, the recognition, the connection to meaning fails to happen. This demonstrates that perception isn't just sensing, but requires higher-level processing that interprets sensory information in light of stored knowledge. Autotopagnosia, difficulty identifying body. Parts can also result from parietal damage. Patients might be unable to point to their elbow when asked, or might point to the wrong body part despite having no sensory or motor deficits. They know what an elbow is conceptually and can point to someone else's elbow, but the connection to their own body representation is disrupted. This reveals that we maintain an internal map of our body, a representation of our physical self that the parietal lobe helps construct and maintain. The parietal lobe also houses neural machinery for mathematical thinking and numerical concepts. The intraparietal sulcus, a groove running through the parietal lobe, shows consistent activation during calculations and number comparison tasks. There's evidence that we have an intuitive number sense, an approximate understanding of quantities, that emerges early in development and depends on parietal function. Even infants show surprise when quantities don't add up as expected, suggesting some innate numerical intuition. This number sense provides the foundation for more sophisticated mathematical thinking. As children learn arithmetic, mathematics education builds upon this intuitive foundation, associating symbolic numbers with quantity representations and learning computational procedures. Brain imaging studies show that mathematical learning involves changes in parietal activity, with practiced calculation showing different patterns than novel ones. Dyscalculia, difficulty learning mathematics, often involves atypical parietal function, supporting the region's importance for numerical cognition. Behind and below the parietal lobe sits the temporal lobe, tucked into the side of the brain, roughly underneath your temples. This lobe handles auditory processing, language comprehension, memory formation, and face recognition, among other functions. The primary auditory cortex, located in the superior temporal gyrus, along the temporal lobe's upper edge, receives information from the ears and begins the process of making sense of sound. It's organized tunotopically, meaning different frequencies of sound activate different locations, creating a spatial map of pitch. High frequencies activate one end of the auditory cortex, low frequencies the other end, with intermediate frequencies mapped in between. This tonotopic organization is maintained throughout the auditory system, from the cochlea in the inner ear, through multiple processing stages in the brainstem and thalamus, to the cortex. From this starting point in primary auditory cortex, sound information flows to surrounding areas where it's processed into increasingly complex representations. Some regions specialize in identifying sound sources, helping you distinguish whether that sound came from in front or behind, from above or below. Others extract speech sounds from background noise, allowing you to follow a conversation in a noisy restaurant. Still others recognize voices, letting you instantly identify a familiar person by their speech alone. The temporal lobe's auditory regions build a rich acoustic landscape, transforming air pressure waves into meaningful auditory experience. Music processing particularly showcases the temporal lobe's sophistication. Different aspects of music activate different regions. Rhythm processing involves motor areas in the basal ganglia, regions associated with timing and movement. Melody processing activates auditory regions in the temporal lobe, with the right hemisphere showing particular sensitivity to melodic contours. Harmony and tonal relationships involve still other regions. Professional musicians show structural differences in these areas, with larger auditory regions and enhanced white matter connections, demonstrating the brain's plasticity in response to extensive musical training. The phenomenon of musical imagery, hearing music in your mind, activates many of the same auditory regions as actually hearing music. When you imagine a melody, your auditory cortex shows activity patterns resembling those evoked by real sound, though typically less intense. This demonstrates that auditory cortex doesn't just respond to sound, but can generate auditory experience internally, creating the subjective experience of hearing even in silence. Wernicke's area, typically in the left hemisphere's superior temporal gyrus, is essential for language comprehension. While Broca's area handles speech production, Wernicke's area handles understanding. Damage here produces receptive aphasia, a condition where speech remains fluent and grammatically structured but becomes essentially meaningless, filled with nonsense words and incoherent ideas. People with Wernicke's aphasia don't realize their speech is nonsensical because they also can't understand others' speech, so they can't detect the mismatch between what they mean to say and what actually comes out. Listen to someone with Wernicke's aphasia and you'll hear speech that sounds superficially normal. The prosody is intact, the rhythm is natural, the grammar is largely preserved, but the content is incomprehensible. They substitute wrong words, create nonsense words called neologisms, and string words together in ways that follow grammatical rules but convey no meaning. It's as if the form of language remains while the meaning has evaporated. The precise boundaries and functions of Wernicke's area remain debated. Some researchers argue it's not a single discrete region, but part of a larger network spanning multiple temporal and parietal areas. Language comprehension involves not just Wernicke's area, but also regions involved in working memory, semantic knowledge, and syntactic processing. Damage anywhere in this network can produce comprehension difficulties, though the specific pattern of impairment varies with lesion location. The temporal lobe's role in memory is even more striking. The medial temporal lobe, the inner surface of the temporal lobe, contains structures critical for memory, particularly the hippocampus and surrounding cortices. We'll discuss the hippocampus in detail when we get to the limbic system, but for now, know that this seahorse-shaped structure is absolutely essential for forming new long-term memories. The famous patient HM, whose real name was Henry Molaison, had his hippocampi removed bilaterally to treat severe epilepsy. The surgery succeeded in reducing his seizures, but left him with devastating memory impairments. He could no longer form new episodic memories. He could meet you, have a conversation, and then 5 minutes later have no recollection of ever seeing you before. His working memory functioned fine. His long-term memories from before the surgery remained intact. But the bridge between short-term and long-term memory had been severed. HM lived for decades after his surgery, and researchers studied him extensively, revolutionizing our understanding of memory. His case demonstrated that memory is not a single system, but involves multiple systems that can be dissociated. HM could still learn new motor skills, showing improvement with practice despite having no memory of practicing. This showed that procedural memory, memory for skills and habits, depends on brain systems separate from the medial temporal lobe. He could also show priming effects, where previous exposure to a stimulus influences processing of that stimulus later, even without conscious memory of the exposure. The temporal lobe also contains the fusiform gyrus, a region on its undersurface that's remarkably specialized for face recognition. This area, sometimes called the fusiform face area, responds much more strongly to faces than to other objects. Brain imaging studies show that when people look at faces, the fusiform gyrus activates robustly. When they look at houses or tools or any other objects, the activation is much weaker. This specialization for faces makes evolutionary sense. Recognizing individuals, reading facial expressions, perceiving age and gender and emotion from faces, all are crucial for social interaction. Humans are intensely social creatures, and faces carry enormous amounts of behaviorally relevant information. Natural selection appears to have shaped dedicated neural machinery for processing this important stimulus category. Damage to the fusiform gyrus can produce prosopagnosia, face blindness, where people lose the ability to recognize faces, even of close family members or their own reflection. They can see faces perfectly well, can describe facial features, can identify people by voice or gait or context. But faces themselves have become meaningless patterns. Some people with prosopagnosia develop compensatory strategies, focusing on distinctive features like hairstyles or glasses or unusual facial characteristics. But they've lost what comes effortlessly to most of us, the immediate recognition that comes from seeing a familiar face. Prosopagnosia can also be developmental, present from birth, affecting roughly 2% of the population. These individuals never develop normal face recognition abilities, though they often don't realize their experience differs from others until well into adulthood. They might avoid social situations where they're expected to recognize people, might seem aloof or unfriendly when they fail to acknowledge acquaintances, might rely heavily on context and non-facial cues. The condition demonstrates that face recognition, which feels so automatic and effortless, depends on specific neural machinery that can fail to develop or can be damaged. The temporal lobe's anterior region, particularly the temporal pole, appears to be involved in semantic memory, your knowledge of facts and concepts. This includes everything from knowing that Paris is the capital of France to understanding what dogs are as a category. Semantic dementia, a degenerative condition that primarily affects the temporal poles, gradually robs people of this conceptual knowledge. People with semantic dementia forget what words mean, what objects are for, what categories things belong to. Show them a picture of a kangaroo and they might not know what it is. They can see it clearly, can describe its features, but the connection to meaning is lost. The word kangaroo becomes just a sound without referent. The syndrome progresses gradually, with more specific concepts lost before more general ones. They might forget kangaroo, but still recognize animal. Eventually, even general concepts erode. What makes semantic dementia particularly poignant is that other cognitive abilities remain relatively preserved. They can still reason logically, can solve problems, can navigate familiar environments. Their episodic memory for recent events is often less impaired than in Alzheimer's disease, but their conceptual knowledge, the shared semantic space that allows communication and understanding, progressively disappears. They become stranded in a world of perceptual experiences that have lost their meaning. Finally, at the back of the brain, we find the occipital lobe, the smallest of the four main lobes but absolutely crucial for vision. The primary visual cortex, also called V1 or the striate cortex, sits at the very back of the occipital lobe wrapped around the calcarine sulcus. This is where visual information from the eyes first reaches the cortex. The journey from photon to conscious visual experience begins in the retina, where photoreceptors convert light into electrical signals. These signals pass through several layers of retinal processing, then travel down the optic nerve to the lateral geniculate nucleus of the thalamus, which relays them to V1. V1 is organized retinotopically, maintaining a map of visual space. Adjacent points in your visual field activate adjacent neurons in V1. But this map is highly distorted. The central region of vision, the fovea where acuity is highest, gets a disproportionately large representation in V1. Roughly half of V1 is devoted to processing the central 10 degrees of vision, even though this represents a tiny fraction of the total visual field. This cortical magnification reflects the retina's structure, with the fovea packed densely with photoreceptors and downstream neurons. V1 doesn't see the way you consciously experience vision. Instead, it breaks the visual world into basic features, edges at particular orientations, contrast changes, motion in specific directions, color information. These are the building blocks from which vision is constructed. Individual neurons in V1 respond selectively to specific features. Some neurons fire only when a vertically oriented edge appears in their receptive field. Others respond to horizontal edges, diagonal edges, edges at any particular angle. Still others respond to direction of motion, to particular colors, to spatial frequencies. This feature detection was revealed in groundbreaking work by David Hubel and Torsten Wiesel, who recorded from individual neurons in cat and monkey visual cortex, showing for the first time how the visual system extracts information from the retinal image. Their work earned them the Nobel Prize and revolutionized our understanding of sensory processing. They showed that vision isn't like a camera simply recording images, but involves active processing, extracting relevant features and constructing representations. From V1, information flows to surrounding visual areas, each specialized for processing particular aspects of vision. V2, immediately adjacent to V1, performs more complex feature analysis, extracting information about figure-ground relationships, depth cues, and more complex shapes. V3 is involved in processing form and motion. V4 shows strong responses to color and is important for color constancy, the ability to perceive colors consistently despite changes in illumination. Area V5, also called MT for middle temporal area, is specialized for motion processing. Damage to V5 produces a striking condition called achinotopsia, motion blindness, where the world appears as a series of static snapshots rather than continuous motion. One famous patient described pouring tea as particularly difficult because she couldn't see the tea rising in the cup, only seeing it stationary at different levels until suddenly it overflowed. Crossing streets became hazardous because cars would appear stationary at one location, then suddenly at another, without the perception of movement between. The condition demonstrates that motion perception isn't just seeing things at different locations at different times, but involves specific neural machinery dedicated to extracting motion information. The brain doesn't build up a single image of the world. Instead, it maintains multiple parallel representations, each emphasizing different aspects of visual information. This parallel processing allows the visual system to extract different types of information simultaneously, making vision both fast and rich in detail. Two major processing streams emerge from the occipital lobe. The ventral stream, flowing forward along the bottom of the brain into the temporal lobe, handles object recognition. This is the what pathway, the system that lets you identify what you're looking at. Neurons in the ventral stream show increasingly complex selectivity as you move forward. Early areas respond to simple shapes. Later areas respond to complex objects like faces, hands, bodies, or specific categories like animals or tools. The dorsal stream, flowing forward along the top of the brain into the parietal lobe, handles spatial information and visual guidance of action. This is the where and how pathway, the system that lets you locate objects in space and guide your movements toward them. Neurons in the dorsal stream encode object locations, motion trajectories, and the relationship between objects and potential actions. These two streams can be selectively damaged, producing distinct syndromes. Damage to the ventral stream produces visual agnosia, the inability to recognize objects by sight despite intact basic vision. Patients can see clearly, can copy drawings, can describe visual features, but can't identify what they're looking at. Show them a picture of a hammer, and they might describe a long thin part and a wider perpendicular part without recognizing it as a hammer. But if they hear it banging or hold it in their hands, recognition is immediate. Damage to the dorsal stream produces optic ataxia, difficulty using vision to guide reaching movements. Patients can identify objects just fine but struggle to reach toward them accurately. Their reaching movements are clumsy and misdirected, as if the connection between seeing where something is and moving toward it has been disrupted. Interestingly, they can often reach accurately towards sounds or toward their own body parts, showing that the deficit is specifically in visually guided action. Damage to the occipital lobe can produce scotomas, blind spots in the visual field. What's fascinating is that people often don't notice these blind spots. The brain fills them in, creating the illusion of continuous vision even where actual visual input is missing. This filling in happens automatically, outside awareness, demonstrating that perception is not passive reception of sensory information, but active construction. Everyone actually has a blind spot in each eye, the location where the optic nerve exits the retina, leaving no photoreceptors. You're not aware of these blind spots because your brain fills them in based on surrounding visual information. You can demonstrate this to yourself with simple diagrams, positioning them so one image falls in the blind spot and watching it disappear. But in everyday life, you never notice the gap because your visual system constructs a seamless experience. More extensive damage can produce cortical blindness, where the eyes function perfectly but the person is blind because the brain can't process visual information. The retina still transduces light into neural signals, the optic nerve still carries those signals, but damage to the visual cortex prevents those signals from producing conscious vision. Some people with cortical blindness develop a phenomenon called blind sight, where they can respond to visual stimuli despite consciously seeing nothing. Blindsight patients report being completely blind in parts of their visual field. Ask them to point to a light, and they'll say they can't because they can't see it. But if you insist they guess, their pointing is often accurate, well above chance. Show them an emotional face, and their physiology shows emotional responses even though they report seeing nothing. They can navigate around obstacles without being aware of seeing them. Some part of their visual system still works, processing information outside conscious awareness. Blindsight reveals that conscious vision is not the same as visual processing. The brain can extract information from visual input, can use that information to guide behavior and emotional responses, without that information reaching consciousness. This dissociation between unconscious processing and conscious awareness raises profound questions about the nature of consciousness and perception. What determines which information becomes conscious? Why do some neural processes produce subjective experience while others don't? These questions remain at the frontier of neuroscience and philosophy. There's a fifth lobe buried deep within the cerebrum, hidden behind the temporal lobe. The insular cortex, or insula, sits inside the lateral sulcus, the deep groove separating the temporal lobe from the frontal and parietal lobes. The insula is involved in a strange mix of functions. It processes taste information and visceral sensations from your internal organs. It's active when you feel disgust, both physical disgust at something revolting and moral disgust at unfair or unethical behavior. It plays a role in awareness of your body's internal state, what neuroscientists call interoception. The insula might be involved in emotions generally and in consciousness itself, though these connections remain debated. It's a small region, easily overlooked, but its diverse functions suggest it might be more important than its size would suggest. Beneath the cerebral cortex lie several subcortical structures, deep brain regions that work in concert with the cortex. The basal ganglia, a collection of nuclei including the caudate nucleus, putamen, and globus pallidus, are critical for selecting and initiating movements, particularly well-learned motor sequences. The basal ganglia also appear to be involved in habit formation, working memory and emotional processing. Parkinson's disease primarily affects the substantia nigra, a structure closely connected to the basal ganglia, leading to the movement difficulties characteristic of the condition. The thalamus, sitting at the top of the brainstem, is sometimes called the brain's relay station. Nearly all sensory information passes through the thalamus on its way to the cortex. Different nuclei within the thalamus relay different types of information: visual, auditory, somatosensory, and so on. But the thalamus does more than just relay. It filters, selecting which information reaches consciousness and which gets screened out. The thalamus also plays roles in regulating consciousness and attention, which is why thalamic damage can produce severe cognitive impairments. The cerebrum, then, is not a single unified structure but a collection of regions, each specialized yet interconnected, each handling particular aspects of perception, thought, and action. These regions don't work in isolation. They form networks, circuits that span multiple areas and even multiple lobes. Understanding the brain isn't just about mapping what each region does, it's about understanding how these regions communicate, how information flows through the system, how distributed networks give rise to unified conscious experience. And that's just the cerebrum. We're not even close to done. Part 2, the cerebellum. The precision engine tucked beneath the occipital lobe, sitting behind the brainstem. The cerebellum is easy to overlook. It's small, accounting for only about 10% of the brain's total volume. But in terms of neurons, the cerebellum is an absolute powerhouse. Despite its small size, it contains more neurons than the rest of the brain combined. Roughly 80% of the brain's neurons are packed into the cerebellum's densely folded structure. If the cerebrum is a sophisticated computer, the cerebellum is a specialized coprocessor, handling specific but crucial computational tasks with remarkable precision. The name cerebellum means little brain, and the resemblance is genuine. Like the cerebrum, the cerebellum has two hemispheres. Like the cerebrum, its surface is covered in grey matter cortex with white matter underneath. Like the cerebrum, it's heavily folded, though the cerebellum's folds are finer and more tightly packed, creating its characteristic layered appearance. When you slice through the cerebellum, the branching pattern of white matter within grey matter creates a pattern called the arbor vitae, Latin for tree of life, because it resembles a branching tree. The cerebellum's computational architecture is remarkably regular. While the cerebrum shows extensive regional specialization, with different areas having different structures and connection patterns, the cerebellar cortex looks essentially the same everywhere. It's organized into 3 layers: the molecular layer on the outside, a thin Purkinje cell layer in the middle, and the granular layer on the inside. This uniform structure suggests that the cerebellum performs the same type of computation regardless of what information it's processing, whether that's motor commands, sensory predictions, or cognitive tasks. This uniformity extends to the cellular level. The cerebellum contains several specific cell types arranged in a highly stereotyped circuit. Granule cells, the most numerous neurons in the brain, are packed densely in the granular layer. Their axons ascend to the molecular layer where they bifurcate, forming parallel fibers that run perpendicular to the Purkinje cell dendrites. Each parallel fiber can contact thousands of Purkinje cells, and each Purkinje cell can receive input from hundreds of thousands of parallel fibers. This creates a matrix of connections that resembles a computer's lookup table, allowing the cerebellum to learn arbitrary input-output mappings. For most of history, the cerebellum was understood primarily as a motor control center. Watch someone with cerebellar damage try to perform coordinated movements, and you'll immediately understand why. They develop ataxia, a loss of coordination that makes even simple movements awkward and imprecise. Reaching for an object becomes a herky-jerky motion, overshooting and then overcorrecting. Walking becomes unsteady, with a wide-based gait that looks almost drunken. Speech becomes slurred and dysarthric, the precise timing of vocal control disrupted. But recent research has revealed that the cerebellum does far more than control movement. It's involved in cognitive functions, in emotional regulation, in virtually any task requiring precise timing or prediction. The cerebellum, it turns out, is not just a motor control center, but a general-purpose prediction and coordination machine. The cerebellum's structure is remarkably uniform. Unlike the cerebral cortex with its regional specialization, the cerebellar cortex looks essentially the same everywhere. It's organized into 3 layers: the molecular layer on the outside, a thin Purkinje cell layer in the middle, and the granular layer on the inside. This uniform structure suggests that the cerebellum performs the same type of computation regardless of what information it's processing, whether that's motor commands, sensory predictions, or cognitive tasks. Purkinje cells are the cerebellum's primary output neurons, and they're among the most distinctive cells in the nervous system. Each Purkinje cell has a massive dendritic tree, branching like a fan in a single plane, that can receive up to 200,000 synaptic inputs. These cells integrate an enormous amount of information and send their outputs to the deep cerebellar nuclei, which then project to other brain areas. Purkinje cells are inhibitory, meaning they decrease activity in their target neurons, allowing the cerebellum to act as a fine-tuning mechanism, sculpting motor and cognitive programs by selectively suppressing unwanted activity. The cerebellum can be divided into 3 functional regions, each with distinct connections and roles. The vestibular cerebellum, the oldest part evolutionarily, includes the flocculonodular lobe and is closely connected to the vestibular system, your sense of balance and spatial orientation. This region helps you maintain your balance, coordinates eye movements with head movements, and keeps you upright against gravity. When you turn your head, the vestibular cerebellum ensures your eyes move to compensate, maintaining visual stability. This vestibulocular reflex is so fast and automatic that you're not consciously aware of it happening. The spinocerebellum, including the vermis and intermediate zones, receives massive inputs from the spinal cord, carrying information about body position and ongoing movements. This region compares the intended movement, the motor plan sent down from the cortex, with the actual movement being executed, as reported by proprioceptive sensors in the muscles and joints. If there's a mismatch, if the movement isn't going as planned, the spinocerebellum sends corrective signals back to the motor cortex and brainstem, adjusting the movement in real time. This is how you can reach accurately for objects, how you can walk across uneven ground without falling, how practiced movements become smooth and fluid. The cerebellum, the largest region and the most recent evolutionary development, includes the lateral hemispheres. This region is connected heavily with the cerebral cortex, particularly the prefrontal and posterior parietal cortices, areas involved in planning, attention, and working memory. The cerebellum doesn't just coordinate movements, it coordinates thoughts and cognitive processes. Evidence suggests it's involved in language, in attention shifting, in mental imagery, in planning sequences of actions, whether physical or abstract. How does the cerebellum work? The leading theory is that it acts as a predictive machine, learning to anticipate the sensory consequences of actions. Every time you move, your action produces sensory feedback, visual changes as your hand enters your field of view, proprioceptive signals as your muscles contract and joints move, tactile sensations if you make contact with something. The cerebellum learns to predict this sensory feedback based on copies of the motor commands. When you reach for a cup, your cerebellum predicts what it should feel like, what it should look like, what sensory information should arrive. If the actual sensory feedback matches the prediction, great. If it doesn't, something unexpected has happened, and the cerebellum adjusts, both correcting the ongoing movement and updating its internal model for future movements. This predictive capability extends beyond movement. The cerebellum appears to predict sensory events even in the absence of action. It's active when you track moving objects with your eyes, predicting where the object will be. It's active during timing tasks, predicting when events will occur. There's evidence that it's involved in predicting what words will come next in a sentence, what notes will come next in a melody, what thoughts will follow in a chain of reasoning. The cerebellum, in this view, is a general-purpose prediction engine, learning the patterns and sequences that characterize both physical and mental events. Motor learning demonstrates the cerebellum's role beautifully. When you learn a new motor skill, whether that's riding a bike or playing a musical instrument or mastering a tennis serve, you're essentially training your cerebellum. Initially, the movements are clumsy and require conscious attention. You have to think about each component, each sub-movement. When learning to ride a bicycle, you consciously think about pedaling, about steering, about balance. You make constant adjustments, overcorrections, your movements jerky and uncertain. But with practice, the cerebellum builds an internal model of the skill, learning the precise timing and sequencing required. The cerebellum receives copies of the motor commands sent from the cortex, along with sensory feedback about the results of those commands. Through repeated practice, it learns to predict the sensory consequences of motor commands. When prediction matches reality, the movement was correct. When there's a mismatch, an error signal triggers learning, adjusting the internal model to improve future performance. Eventually, the movement becomes automatic, flowing without conscious thought. You no longer think about how to balance on the bicycle. You just ride. The cerebellum has taken over, executing the learned program with a precision that conscious control could never achieve. This transition from effortful to automatic reflects changes in the brain. Initially, prefrontal and parietal cortices show high activity as you consciously control the movement. With practice, activity shifts to the cerebellum and motor cortex, with reduced prefrontal involvement. The skill has become proceduralized, freeing conscious resources for other tasks. This learning mechanism appears to involve long-term depression at the synapses between parallel fibers and Purkinje cells. When a parallel fiber is active at the same time as climbing fiber input, which signals error, the synapse weakens. Over many repetitions, this selective weakening shapes the Purkinje cell's response pattern, tuning it to produce the desired output. The cerebellum essentially learns through trial and error, using error signals to gradually refine its internal models. The cerebellum's role in motor adaptation extends beyond skill learning to constant ongoing adjustments. When you put on a new pair of glasses with different magnification, the world initially appears distorted. Reaching for objects feels wrong because the visual information doesn't match your learned expectations. But within hours to days, your cerebellum adapts. It recalibrates the mapping between visual input and motor output, and reaching becomes accurate again. Remove the glasses, and you experience negative after effects as the cerebellum readapts to normal vision. This adaptive capability allows us to maintain accurate motor control despite changes in our body, our tools, or the environment. As you grow, as your limbs lengthen and your strength changes, your cerebellum continuously updates its models. When you use a tool, extending your effective reach, your cerebellum learns the tool's properties and incorporates it into your body schema. Master craftspeople and athletes develop such finely tuned cerebellum models that their movements appear effortless, their tools seeming like natural extensions of their body. Classical conditioning, one of the most basic forms of learning, depends critically on the cerebellum. The classic example is eye blink conditioning. Pair a tone with a puff of air to the eye, and eventually the tone alone will produce a blink. This learned response requires the cerebellum. Lesion-specific regions of the cerebellar cortex or the deep cerebellar nuclei, and eye blink conditioning fails completely. The cerebellum learns the timing, learning to produce the blink at just the right moment to protect the eye from the anticipated air puff. This might seem like a simple reflex, but it demonstrates a fundamental computational capability, learning to predict and time responses based on experience. The cerebellum receives information about the tone, about the air puff, about the timing relationship between them. Through repeated pairings, it learns that the tone predicts the puff, and it generates a precisely timed blink response. The timing is crucial. Blink too early or too late, and the puff reaches the eye. The cerebellum learns to produce the blink with millisecond precision, demonstrating its extraordinary temporal processing capabilities. Eye blink conditioning generalizes to more complex learning. Any situation requiring precise timing of motor or cognitive responses likely involves the cerebellum. Speech production requires exquisite timing of vocal cord movements, tongue positions, lip configurations. The cerebellum helps coordinate these movements, producing the flowing speech we take for granted. Disruption of cerebellar timing produces dysarthria, speech that's slow, irregular, and difficult to understand. Musical performance provides another example of the cerebellum's timing capabilities. Playing an instrument requires precise temporal coordination of numerous movements. Each note must be the correct duration, must transition to the next note with precise timing. In ensemble playing, your timing must coordinate with other musicians, requiring prediction of their actions. Professional musicians show enhanced cerebellar activation during performance, and cerebellar damage impairs musical timing and rhythm. The cerebellum's timing abilities extend far beyond motor control. Rhythm perception, whether in music or speech, depends on cerebellar function. People with cerebellar damage often struggle with rhythm, both perceiving it and producing it. They can't tap along to a beat with normal accuracy. They can't detect subtle timing deviations in melodies. Their speech loses its natural prosody, the rhythmic and melodic contours that convey emotion and emphasis. There's growing evidence that the cerebellum plays a role in autism spectrum disorders. Many individuals with autism show structural abnormalities in the cerebellum, and cerebellar-like symptoms, including motor coordination difficulties and timing problems, are common. Some researchers hypothesize that impaired cerebellar prediction might contribute to the sensory and social difficulties in autism, though this remains speculative. If the cerebellum helps predict sensory consequences of actions and events in the world, then impaired prediction could make the world seem more chaotic and less predictable, potentially contributing to the preference for routine and resistance to change often seen in autism. The cerebellum's role in cognition extends to language processing. Functional imaging studies show cerebellar activation during various language tasks: reading, speaking, listening to speech, even silent word generation. The right cerebellum, which connects to the left language-dominant cerebral cortex, shows particular involvement in language. Cerebellar damage can produce language difficulties that aren't simply motor speech problems, but appear to involve higher-level language processing. One theory suggests the cerebellum acts as a timing and sequencing device for language, just as it does for movement. Language requires precise temporal coordination. Speech sounds must be produced in the correct sequence with appropriate timing. Understanding speech requires tracking rapidly changing acoustic information over time. Reading involves coordinating eye movements with visual processing and phonological decoding. All these processes involve timing and sequence, domains where the cerebellum excels. The cerebellum also appears involved in attention and working memory. The cerebellum connects extensively with prefrontal cortex, the brain's main executive control center. These connections allow the cerebellum to influence cognitive control processes. Some researchers propose that the cerebellum performs error detection and correction, not just for motor actions but for cognitive operations, helping to maintain attention, detect mistakes in reasoning, and optimize cognitive strategies. Cerebellar cognitive affective syndrome, described in patients with cerebellar lesions, includes impairments in executive function, working memory, and language, along with personality changes like flattened affect and inappropriate behavior. The syndrome is most common with lesions to the posterior lobe of the cerebellum, the region most heavily connected to frontal and parietal cortices. This demonstrates that the cerebellum is not just a motor structure but an integral part of Cognitive and emotional processing. The specific cognitive symptoms depend on which part of the cerebellum is damaged. The posterior lateral cerebellum connected to prefrontal cortex appears particularly important for cognition. Damage here produces executive dysfunction, difficulty with planning, working memory, and set shifting. The posterior vermis, connected to limbic structures, shows involvement in emotional regulation. Damage produces emotional instability, sometimes inappropriate affect. The anterior cerebellum, connected to motor cortex, remains primarily involved in motor control. This topographic organization reflects the cerebellum's role as a general-purpose coordination device. Different cerebellar regions coordinate different aspects of brain function, but the computational algorithm appears similar throughout. The cerebellum receives input, builds predictive models, detects errors, and generates corrective signals, whether for motor control, cognitive processing, or emotional regulation. Cerebellar damage produces distinctive symptoms. Beyond the motor coordination we've mentioned, people often develop dysmetria, an inability to judge distances, causing them to overshoot or undershoot when reaching for objects. They develop intention tremor, shaking that emerges specifically during voluntary movement, getting worse as they approach their target. They develop dysdiadochokinesia, difficulty performing rapid alternating movements, like flipping the hand back and forth. Their speech becomes scanning or explosive, losing its natural rhythm and flow. Gait ataxia, instability during walking, is particularly common. People walk with a wide-based gait, legs far apart for stability, and still tend to veer to one side. Tandem gait, walking heel to toe in a straight line, becomes impossible. The Romberg test, where you stand with feet together and eyes closed, reveals instability even during standing. These motor symptoms reflect the cerebellum's fundamental role in coordination and balance. But cerebellar patients also show cognitive changes. They may have difficulty with tasks requiring mental timing and sequencing. They may show reduced verbal fluency, struggling to generate words in a category. They may have trouble with tasks requiring rapid switching between mental sets. These cognitive symptoms are often subtle but can significantly impact daily functioning, especially when combined with the motor impairments. Cerebellar disorders have numerous causes. Stroke affecting the cerebellar arteries can produce sudden cerebellar dysfunction. Tumours in or near the cerebellum can compress and damage cerebellar tissue. Multiple sclerosis often affects cerebellar white matter, disrupting connections. Chronic alcohol abuse is particularly toxic to the cerebellum, especially Purkinje cells, leading to progressive ataxia that may only partially recover with abstinence. Spinocerebellar ataxias are a group of genetic disorders causing progressive cerebellar degeneration. Different genetic mutations produce different patterns of symptom progression and severity, but all involve gradual loss of cerebellar function. Symptoms typically begin in adulthood with mild incoordination that progressively worsens. Eventually walking becomes impossible, fine motor tasks like writing become severely impaired, speech becomes incomprehensible. The progressive nature makes these disorders particularly devastating, as people watch their motor and cognitive abilities slowly deteriorate. Cerebellar hypoplasia, underdevelopment of the cerebellum, can occur congenitally. In some cases, the cerebellum may be nearly absent, a condition called cerebellar agenesis. Remarkably, some individuals with severe cerebellar hypoplasia develop relatively normally, suggesting remarkable brain plasticity. Other structures may partially compensate for cerebellar loss, though subtle motor and cognitive impairments typically remain. The fact that some compensation is possible highlights that the brain is not a collection of independent modules, but a network where other regions can sometimes take over functions when one region is damaged. The cerebellum receives input from virtually the entire brain and sends its outputs back to motor and cognitive control centers. It sits in a position to monitor and modulate almost everything the brain does. It doesn't initiate actions or thoughts, but it refines them, makes them more precise, more appropriately timed, more coordinated. Without the cerebellum, you could still move, still think, still feel emotions, but everything would be clumsier, less coordinated, less smoothly integrated. And perhaps that's the best way to understand the cerebellum. It's not the conductor of the brain's orchestra, but it is the section that keeps time, that ensures each instrument comes in precisely when it should, that maintains the rhythm, that allows the whole ensemble to function as a unified whole rather than a cacophony of individual parts. Part 3. The brainstem, the foundation of life. If you removed the cerebrum and cerebellum entirely, what would remain is the brainstem, a stalk-like structure extending down from the base of the brain to merge with the spinal cord. The brainstem is ancient, evolutionarily speaking. Every vertebrate has one. Fish have brainstems, reptiles have brainstems, birds have brainstems. The basic structure has been conserved across hundreds of millions of years of evolution because it performs functions absolutely essential to life. The brainstem is small, only about 3 inches long, and making up less than 3% of the brain's total weight. But damage to the brainstem is often catastrophic, in ways that damage to other brain regions is not. You can lose significant amounts of cerebral cortex and remain alive, functional, even conscious. Lose your brainstem, and you stop breathing. Your heart stops beating. Consciousness vanishes. The brainstem maintains the autonomic functions that keep you alive without any conscious effort on your part. The brainstem divides into 3 main regions, from top to bottom: the midbrain, the pons, and the medulla oblongata. Each region has its own specific structures and functions, though they work together seamlessly to control vital functions and relay information between the brain and body. The midbrain, or mesencephalon, is the uppermost portion of the brainstem, sitting at the base of the forebrain. Despite being the smallest of the 3 brainstem divisions, it contains several critical structures. The tectum, Latin for roof, forms the dorsal portion of the midbrain. It contains 4 bumps called the colliculae. 2 superior colliculae involved in visual processing, and 2 inferior colliculae involved in auditory processing. The superior colliculae don't handle conscious vision, but instead control reflexive eye movements, the automatic shifts of gaze toward moving or novel stimuli. When something moves in your peripheral vision and your eyes automatically flick toward it, that's your superior colliculae at work. They create a map of visual space and generate the motor commands to point your eyes, and to some extent your head, toward interesting locations. The superior colliculus integrates multiple types of information to guide these orienting responses. It receives direct visual input from the retina, bypassing the cortex entirely. This allows for extremely fast responses to visual events, faster than cortical processing would permit. It also receives input from auditory and somatosensory systems, allowing it to orient towards sounds or touch as well as visual stimuli. The superior colliculus essentially creates a multisensory map of space, with different sensory modalities aligned so that visual, auditory and tactile information from the same location in space converge on the same neurons. This multisensory integration enhances detection and localization. If you hear a sound and see movement from the same location simultaneously, the superior colliculus responds more strongly than to either stimulus alone. This multisensory enhancement makes sense evolutionarily. A predator approaching might produce both sounds and visual movement, and being able to detect and orient toward multiple convergent cues provides survival advantage. The superior colliculus also plays a role in attention. It doesn't just generate eye movements, but influences which locations in space receive enhanced processing throughout the visual system. When the superior colliculus signals that a location is important, visual processing of that location is enhanced even before the eyes move there. This allows the brain to begin processing important information before the high-resolution fovea is directed toward it. The inferior colliculi are way stations in the auditory pathway, receiving information from the auditory brainstem nuclei below and sending it upward to the thalamus and eventually the auditory cortex. They're not just passive relays but perform important processing. The inferior colliculi are particularly involved in temporal processing of sound, detecting gaps, changes and patterns in auditory input. They also participate in sound localization, helping you determine where sounds are coming from based on tiny timing and intensity differences between your 2 ears. Sound localization relies on 2 main cues: interaural time differences, the difference in when a sound reaches each ear, are particularly important for low-frequency sounds. A sound coming from the left reaches the left ear slightly before the right ear. The inferior colliculus contains neurons sensitive to these timing differences, with different neurons tuned to different delays. Interaural intensity differences, the difference in loudness between the 2 ears, are particularly important for high-frequency sounds. The head creates a sound shadow, making sounds quieter in the ear farther from the source. The inferior colliculus integrates these cues to compute sound source location. This happens fast, automatically, without conscious thought, allowing you to orient towards sound sources even before you consciously register what you're hearing. When you hear a sudden noise behind you and immediately turn toward it, that reflexive orienting involves the inferior colliculus working with the superior colliculus to coordinate auditory visual attention. Below the tectum lies the tegmentum, Latin for covering, which contains several important nuclei. The periaqueductal grey, a region of grey matter surrounding the cerebral aqueduct, is involved in pain modulation. This region contains high densities of opioid receptors and produces profound analgesia when stimulated. The periaqueductal grey is part of the descending pain control system, a network of structures that can modulate pain perception by inhibiting pain signals at the level of the spinal cord. This endogenous pain control system explains phenomena like stress-induced analgesia, where people injured in traumatic situations don't feel pain until later. The periaqueductal grey, activated by stress, suppresses pain signaling, allowing people to escape danger without being incapacitated by pain. Morphine and other opioid drugs work partly by activating this system, mimicking the body's natural pain control mechanisms. The periaqueductal grey is also involved in defensive behaviours, coordinating the behavioural and autonomic responses to threats. Different regions of the periaqueductal grey control different defensive strategies. The dorsal region is involved in active defence, producing flight or fight responses. The ventral region is involved in passive defensive responses like freezing. These responses are accompanied by appropriate autonomic changes, cardiovascular adjustments, respiratory changes, and hormonal responses that support the behavioural response. The substantia nigra, a dark pigmented nucleus rich in dopamine-producing neurons, is part of the basal ganglia system despite being located in the midbrain. The dark pigmentation comes from neuromelanin, a byproduct of dopamine metabolism that accumulates in these neurons over time. These dopaminergic neurons are crucial for voluntary movement and reward processing. They project to the striatum, forming the nigrostriatal pathway that's essential for initiating and controlling movement. In Parkinson's disease, these neurons degenerate, and as their numbers decline, the motor symptoms of Parkinson's emerge: tremor at rest, rigidity, bradykinesia, meaning slowness of movement, and postural instability. Symptoms don't appear until roughly 80% of the substantia nigra neurons have been lost, demonstrating the system's remarkable ability to compensate for gradual damage. But once symptoms appear, they progressively worsen as more neurons die. L-DOPA, the main medication for Parkinson's, works by providing a chemical precursor that the remaining substantia nigra neurons can convert to dopamine, temporarily alleviating symptoms. But L-DOPA doesn't stop the disease process, and as more neurons die, higher doses are needed, eventually leading to diminishing returns and problematic side effects. Deep brain stimulation, where electrodes are implanted in the subthalamic nucleus or globus pallidus, can help some patients by modulating basal ganglia activity, partially compensating for dopamine loss. The substantia nigra isn't just involved in movement. These dopamine neurons also respond to reward and reward-predicting cues. When something unexpectedly good happens, substantia nigra neurons show a burst of activity, signaling positive prediction error. This dopamine signal is thought to be crucial for reinforcement learning, for learning which actions lead to rewarding outcomes. The nigrostriatal pathway helps translate these reward signals into motivated behaviour, biasing action selection toward rewarding options. The red nucleus, despite its name not actually red in life, is involved in motor coordination, particularly of limb movements. It receives input from the cerebellum and motor cortex, and sends output to the spinal cord via the rubrospinal tract, one of the descending motor pathways. In humans, the rubrospinal tract is relatively small compared to other mammals, as the much larger corticospinal tract has largely taken over its functions. But the red nucleus remains important for coordinating reaching movements and for postural adjustments during movement. The ventral tegmental area, another cluster of dopaminergic neurons, is heavily involved in reward, motivation, and addiction. These neurons project to the nucleus accumbens in the ventral striatum and to the prefrontal cortex, forming the mesocortico-limbic pathway, sometimes called the reward pathway. This system is activated by natural rewards like food and sex, by drugs of abuse, and by cues predicting rewards. The ventral tegmental area's dopamine neurons don't simply signal pleasure, rather they signal reward prediction error, the difference between expected and actual reward. When a reward is better than expected, these neurons fire strongly. When a reward is worse than expected, they decrease their activity below baseline. When a reward is exactly as expected, they don't respond to the reward itself, but may respond to cues predicting it. This prediction error signal is thought to drive learning, adjusting predictions to match reality and guiding future behavior. Drug addiction involves pathological changes in this system. Drugs of abuse typically cause massive dopamine release, far exceeding natural rewards. The brain learns these drug-associated cues extremely strongly, creating powerful motivations to seek drugs. With repeated use, the system adapts, becoming less sensitive to dopamine, requiring more drug to achieve the same effect, and causing withdrawal symptoms when the drug is absent. The ventral tegmental area and its projections undergo structural and functional changes that persist long after drug use stops, contributing to the chronic relapsing nature of addiction. The midbrain also contains the nuclei for cranial nerves 3 and 4. The oculomotor nerve, cranial nerve 3, controls most eye movements and pupil constriction. Damage here produces a characteristic presentation. The affected eye points down and outward, the eyelid droops and the pupil is dilated. The trochlear nerve, cranial nerve 4, controls the superior oblique muscle, which rotates the eye downward and inward. It's the only cranial nerve to emerge from the back of the brainstem, and damage produces double vision, especially when looking down and inward, making activities like reading or descending stairs difficult. Below the midbrain sits the pons, Latin for bridge. The pons is a bulge on the front of the brainstem, and its name reflects its role as a connection point between various brain regions. The pons is continuous with the midbrain above and the medulla below, and posteriorly it connects to the cerebellum via the massive middle cerebellar peduncles. The anterior or ventral surface of the pons shows a characteristic bulge, formed by the transverse pontocerebellar fibers. These fibers wrap around the brainstem, creating the prominent bulge visible from the outside. The basilar groove runs down the midline of this ventral surface, and it's where the basilar artery sits, the major blood vessel supplying the pons and nearby structures. The pons can be divided into two major regions functionally and anatomically. The ventral pons, the bulging anterior portion, contains the pontine nuclei. These nuclei receive massive input from the cerebral cortex, particularly from motor and association areas. Virtually the entire cerebral cortex sends projections to the pontine nucle, providing information about ongoing cortical activity. The pontine nuclei then relay this information to the cerebellum via the middle cerebellar peduncles, which contain over 20 million nerve fibers, making them the largest of the 3 cerebellar peduncles. This cerebropontocerebellar pathway is crucial for cerebellar function. The cerebellum needs to know what the cerebral cortex is planning and doing to generate appropriate predictions and corrections. When the motor cortex sends commands to move, copies of those commands go to the pontine nuclei and then to the cerebellum, allowing the cerebellum to prepare for the sensory consequences of the movement and to make any necessary adjustments in real time. The dorsal pons, or the pontine tegmentum, contain structures that are evolutionarily older and functionally distinct from the ventral pons. The tegmentum is the evolutionarily older part of the pons, part of the reticular formation that extends throughout the brainstem. This region contains various nuclei involved in arousal, sleep, respiratory control, and other vital functions. The pons contains several cranial nerve nuclei. The trigeminal nerve, cranial nerve 5, and the largest cranial nerve has multiple nuclei spread across the pons and extending into the medulla and midbrain. The main sensory nucleus, located in the mid-pons, receives touch and proprioceptive information from the face. The mesencephalic nucleus, extending up into the midbrain, receives proprioceptive information from the jaw muscles. The motor nucleus controls the muscles of mastication, the chewing muscles. The spinal trigeminal nucleus, extending down into the medulla, processes pain and temperature information from the face. Trigeminal neuralgia, an extremely painful condition affecting the trigeminal nerve, produces sudden, severe facial pain, often described as like electric shocks. The pain can be triggered by simple activities like touching the face, eating, or even a breeze. The The condition illustrates the importance of the trigeminal nerve for facial sensation and shows how devastating it can be when this system malfunctions. Treatment often involves medications that reduce nerve excitability, or, in severe cases, surgical procedures to decompress the nerve or lesion parts of the trigeminal pathway. The abducens nerve, cranial nerve 6, has its nucleus in the caudal pons. This nerve controls the lateral rectus muscle, which turns the eye outward. The abducens nucleus has an unusual function beyond just controlling the lateral rectus. It also contains interneurons that project to the oculomotor nucleus in the midbrain, coordinating eye movements so both eyes move together. When you look to the right, your right eye's lateral rectus must contract, while your left eye's medial rectus contracts. The abducens nucleus helps coordinate this conjugate gaze, ensuring both eyes move in tandem. Damage to the abducens nerve or nucleus produces a characteristic pattern. The affected eye cannot turn outward, causing double vision. That's worse when looking toward the affected side. The eye may drift inward due to unopposed action of the medial rectus. This is one of the more common cranial nerve palsies, occurring with increased intracranial pressure, trauma, or vascular problems affecting the pons. The facial nerve, cranial nerve 7, has its nucleus in the caudal pons, though the nerve itself exits at the pontomedullary junction. The facial nucleus controls the muscles of facial expression, all those tiny muscles that allow you to smile, frown, raise your eyebrows, scrunch your nose. These movements might seem trivial, but they're crucial for human communication. Facial expressions convey emotions, social signals, intent. Loss of facial expression, as occurs in Parkinson's disease or with facial nerve damage, profoundly impairs social interaction. The facial nerve's path through the brain is unusual. The motor fibers don't exit the brainstem directly from the facial nucleus. Instead, they loop around the abducens nucleus before exiting, a path called the internal genu of the facial nerve. This creates an anatomical landmark useful for localizing brainstem lesions. Damage to the facial nucleus or nerve produces peripheral facial palsy, affecting the entire half of the face. Damage to the motor cortex or its descending pathways produces central facial palsy, affecting only the lower face because the upper face receives bilateral cortical input. Bell's palsy, a common condition causing sudden facial weakness, results from facial nerve inflammation or compression. The cause is often unclear, though viral infections may trigger some cases. One side of the face suddenly becomes weak, causing drooping, difficulty closing the eye, and changes in taste on the front 2/3 of the tongue. Most cases gradually recover, though some people have permanent weakness. The condition illustrates how dependent we are on facial nerve function for normal facial movement and expression. The vestibulocochlear nerve, cranial nerve 8, enters the brainstem at the pontomedullary junction, though its nuclei are located in both the pons and medulla. This nerve has two distinct components. The cochlear division carries hearing information from the cochlea in the inner ear. The vestibular division carries balance and spatial orientation information from the vestibular organs in the inner ear. The cochlear nuclei in the lower pons receive auditory information and send it to the superior olivary complex, also in the pons, where sound localization begins, and then upward to the inferior colliculus. The vestibular nuclei receive information about head position and movement. These nuclei have extensive connections, sending information to the cerebellum, to motor nuclei controlling eye movements, and down to the spinal cord to control posture and balance. The vestibular system is remarkable in its ability to maintain stable vision during head movement and stable posture during movement and perturbations. The vestibulo-ocular reflex, mediated by connections between vestibular nuclei and eye movement nuclei, keeps your eyes stable when your head moves. When you turn your head right, your eyes automatically move left by an equal amount, keeping your gaze fixed on a target. This reflex is so fast and automatic that you're never aware of it, yet it's essential for clear vision during movement. Damage to the vestibular system produces vertigo, a sense that the world is spinning, along with nausea, imbalance and nystagmus, involuntary rhythmic eye movements. Vestibular neuritis, inflammation of the vestibular nerve, produces sudden severe vertigo that gradually improves over days to weeks as the brain compensates for the lost vestibular input. Meniere's disease, affecting the inner ear, produces episodic vertigo along with hearing loss and tinnitus. These conditions demonstrate how much we depend on the vestibular system, even though we're not consciously aware of its constant activity. The reticular formation extends through the pons, a diffuse network of neurons forming the core of the brainstem. This might be one of the most important structures in the brain, despite not being a discrete, clearly bounded region. The reticular formation is involved in arousal and consciousness, in regulating sleep-wake cycles, in modulating sensory information, in controlling certain autonomic functions. The ascending reticular activating system, formed by reticular formation neurons that project upward to the thalamus and cortex, is what keeps you awake and alert, maintaining the level of consciousness necessary for the cortex to process information. Damage to the reticular formation can produce coma, a state of complete unresponsiveness from which the person cannot be awakened. The reticular formation essentially sets the gain on cortical processing, determining how responsive the cortex is to sensory input. This arousal system doesn't function uniformly throughout the day. It shows circadian rhythms, being more active during expected wake times and less active during expected sleep times. It responds to stimuli, increasing arousal when important or novel stimuli appear. It's modulated by various neurotransmitter systems, including noradrenergic neurons in the locus coeruleus, serotonergic neurons in the raphenuclei, cholinergic neurons in the tegmentum, and histaminergic neurons in the hypothalamus. These systems work together to regulate arousal and sleep-wake states. The pons also contains respiratory control centers that work in conjunction with those in the medulla. The pneumataxic center in the upper pons helps regulate the breathing rate and pattern, influencing the transition from inspiration to expiration. The apneustic center in the lower pons stimulates inspiration, promoting deep, prolonged inspiratory efforts. These centers receive input from chemoreceptors, monitoring carbon dioxide and oxygen levels, and from stretch receptors in the lungs, adjusting breathing automatically to maintain appropriate blood gas levels. The integration of respiratory control across the pons and medulla demonstrates the brainstem's sophistication. Breathing isn't a simple reflexive process, but involves multiple control centers coordinating to produce appropriate respiratory patterns that can adapt to changing metabolic demands, voluntary control during speech or breath holding, and reflexive responses to chemical changes or airway irritation. The medulla oblongata, often just called the medulla, is the lowest portion of the brainstem, continuous with the spinal cord below. In many ways, it's the most critical brainstem structure because it houses control centers for the most vital autonomic functions. The medulla is roughly pyramid-shaped, narrowing as it transitions into the spinal cord. On its anterior surface you can see the pyramids, prominent bulges formed by the corticospinal tracts carrying motor commands from the cortex to the spinal cord. At the bottom of the medulla, roughly 80 to 90% of these fibers cross to the opposite side in the pyramidal decussation. This crossing explains why the right motor cortex controls the left side of the body and vice versa. The remaining 10 to 20% of fibers continue on the same side, forming the anterior corticospinal tract. The medulla contains the cardiovascular center, clusters of neurons that regulate heart rate and blood pressure. These neurons are not anatomically discrete nuclei, but are distributed throughout the medullary reticular formation. Some neurons increase heart rate and blood pressure, others decrease them. The balance of activity in these populations determines cardiovascular output. The cardiovascular center receives input from baroreceptors in the carotid sinus and aortic arch, specialized sensors that detect blood pressure changes. When blood pressure rises, the baroreceptors fire more rapidly, signaling the medulla to decrease heart rate and dilate blood vessels, lowering blood pressure. When blood pressure falls, baroreceptor firing decreases, triggering increases in heart rate and vasoconstriction. This baroreflex operates continuously, moment to moment, maintaining blood pressure within appropriate ranges without any conscious awareness or effort. The cardiovascular center also receives input from chemoreceptors, detecting blood oxygen and carbon dioxide levels. When oxygen drops or carbon dioxide rises, the medulla increases cardiovascular output, ensuring adequate oxygen delivery to tissues. The medulla integrates multiple signals, balancing competing demands to maintain appropriate blood pressure and perfusion despite changing conditions. Physical or emotional stress activates the cardiovascular center through connections with the hypothalamus and limbic system. When you're threatened or anxious, your heart rate and blood pressure increase, preparing you for action. When you're relaxed and calm, they decrease. The medulla translates these brain states into appropriate cardiovascular adjustments. The medulla also contains the respiratory center, which generates the basic rhythm of breathing. Even when you're unconscious, even when you're not thinking about breathing at all, these neurons fire rhythmically, sending signals to the diaphragm and intercostal muscles, maintaining the regular cycle of inhalation and exhalation that keeps you alive. The respiratory rhythm generation involves complex interactions between multiple neuronal populations in the medulla. The pre-Butzinger complex, a small region in the ventral medulla, appears to be the primary rhythm generator. Neurons here show intrinsic rhythmic bursting activity, and damaging this region abolishes rhythmic breathing. The rhythm then propagates to other medullary regions that shape the breathing pattern and coordinate the activation of different respiratory muscles. The respiratory center responds primarily to carbon dioxide levels in the blood. As carbon dioxide rises, which happens continuously as cells metabolize nutrients, breathing rate and depth increase, expelling more carbon dioxide and bringing in more oxygen. As carbon dioxide falls, breathing slows. This feedback system maintains blood gas levels within narrow ranges essential for cellular function. Carbon dioxide dissolves in blood to form carbonic acid, affecting blood pH, so respiratory control is also pH control. Interestingly, the respiratory center is relatively insensitive to low oxygen under normal circumstances. The body prioritizes carbon dioxide removal over oxygen intake, which seems counterintuitive. But this makes sense because small changes in oxygen saturation don't have immediate consequences, while carbon dioxide accumulation quickly becomes problematic. Peripheral chemoreceptors in the carotid bodies and aortic bodies do respond to low oxygen, providing a backup system that increases breathing when oxygen levels drop dangerously low. Voluntary control can override the automatic respiratory rhythm. You can choose to breathe faster or slower, can hold your breath, can breathe deeply or shallowly. This voluntary control originates in the motor cortex and bypasses the medullary respiratory center. But the automatic system always wins eventually. Hold your breath long enough, and rising carbon dioxide levels will eventually trigger an irresistible urge to breathe. You cannot voluntarily stop breathing to the point of unconsciousness, because loss of consciousness means loss of voluntary control, and the automatic system immediately resumes breathing. The medulla houses several reflexes essential for survival. The cough reflex, triggered by irritation in the airways, produces a forceful expulsion of air to clear the obstruction. Receptors in the airways detect irritation and send signals to the medulla. The medulla coordinates a complex sequence: deep inspiration, closure of the vocal cords to build pressure, then sudden opening of the vocal cords while abdominal and intercostal muscles contract forcefully, producing the explosive exhalation of a cough. This reflex helps protect the airways from harmful substances and helps clear mucus and debris. The gag reflex, triggered by stimulation of the back of the throat, prevents choking by initiating swallowing or triggering protective responses. Touch the back of the throat and muscles contract to close off the airway, while either pushing the stimulus back out or initiating swallowing to move it down. This reflex involves the glossopharyngeal and vagus nerves, which carry sensory information from the throat to the medulla, and the medulla coordinates the motor response. The vomiting center in the medulla coordinates the complex sequence of events that produces vomiting. Vomiting protects the body from ingested toxins, though it can also be triggered by motion sickness, vestibular dysfunction, or various medical conditions. The sequence involves nausea, salivation to protect teeth from stomach acid, deep inspiration, closure of the glottis, relaxation of the upper gastrointestinal sphincters, and forceful contraction of abdominal muscles and diaphragm. This coordinated sequence requires precise timing and involves multiple muscle groups, all orchestrated by the medullary vomiting center. Swallowing, despite seeming simple, is a remarkably complex process requiring precise coordination of numerous muscles. The medulla contains the swallowing center that coordinates this process. Swallowing progresses through 3 phases. The oral phase is voluntary, where the tongue pushes food toward the back of the mouth. The pharyngeal phase is reflexive, involving elevation of the soft palate to close off the nasal cavity, closure of the vocal cords to protect the airway, and coordinated contractions of pharyngeal muscles to move food through the pharynx. The esophageal phase involves peristaltic waves moving food down the esophagus to the stomach. Damage to the medullary swallowing center produces dysphagia, difficulty swallowing. This can be devastating because it prevents normal eating and drinking and creates a risk of aspiration, where food or liquid enters the airways instead of the esophagus. Aspiration can cause pneumonia, a serious complication. Dysphagia is common after strokes affecting the brainstem and in neurodegenerative diseases affecting the medulla. Several cranial nerves have their nuclei in the medulla. The glossopharyngeal nerve, cranial nerve 9, is involved in taste from the posterior third of the tongue, sensation from the throat and middle ear, and control of swallowing muscles. It also carries sensory information from the carotid body, involved in sensing blood oxygen levels, and from the carotid sinus, involved in sensing blood pressure. The glossopharyngeal nerve thus integrates multiple functions: sensation, taste, motor control, and autonomic sensing. The vagus nerve, cranial nerve 10, is a wanderer. Vagus means wandering in Latin, sending branches throughout the thorax and abdomen. It's the main nerve of the parasympathetic nervous system, the rest and digest system. The vagus nerve slows heart rate, stimulates digestive activity, and influences numerous other autonomic functions. It carries sensory information from the thoracic and abdominal organs back to the brain, providing information about the body's internal state. It controls muscles of the pharynx and larynx involved in swallowing and speech. The vagus nerve's extensive distribution means it influences an extraordinary range of functions. Vagal tone, the baseline level of vagus nerve activity, correlates with various health outcomes. High vagal tone is associated with better emotional regulation, better stress resilience, better cardiovascular health. Interventions that increase vagal tone, including exercise, meditation, and deep breathing, may improve these outcomes. Vagal nerve stimulation, where an implanted device electrically stimulates the vagus nerve, is used to treat epilepsy and depression, demonstrating the nerve's influence on brain function. The accessory nerve, cranial nerve 11, has both cranial and spinal portions. The cranial portion arises from the medulla and joins the vagus nerve, contributing to control of pharyngeal and laryngeal muscles. The spinal portion arises from the upper spinal cord and controls the sternocleidomastoid and trapezius muscles, which move the head and shoulders. Damage to the accessory nerve produces weakness, turning the head away from the affected side and difficulty shrugging the shoulder on the affected side. The hypoglossal nerve, cranial nerve 12, controls tongue movements essential for speech and swallowing. The tongue is remarkably mobile and versatile, capable of complex three-dimensional movements. This mobility depends on intrinsic tongue muscles that change the tongue's shape and extrinsic muscles that move the tongue as a whole. All these muscles are controlled by the hypoglossal nerve. Damage to the hypoglossal nerve or nucleus produces tongue weakness on the affected side. The tongue deviates toward the affected side when protruded, because the unaffected side pushes more strongly. Speech becomes slurred, particularly sounds requiring precise tongue positioning like L, T, and D. Swallowing becomes difficult because the tongue can't properly move food. Bilateral hypoglossal damage can be life-threatening because the tongue may obstruct the airway. The medulla also contains important sensory relay nuclei. The dorsal column nuclei, including the gracile nucleus and cuneate nucleus, receive sensory information about fine touch, vibration sense, and proprioception from the body via the dorsal columns of the spinal cord. These nuclei process this information and relay it to the thalamus via the medial lemniscus, a major sensory pathway running through the brainstem. The spinal trigeminal nucleus, extending from the pons through the medulla, processes pain and temperature information from the face. The different parts of this elongated nucleus process information from different parts of the face and have somewhat different functions. The caudal part, in the lower medulla, processes pain and temperature most strongly. Damage here can produce loss of pain and temperature sensation on the face, while preserving light touch, a dissociated sensory loss that helps localize lesions. The inferior olivary nucleus, a prominent structure in the medulla visible as an olive-shaped bulge on its lateral surface, projects to the cerebellum and plays a role in motor learning and timing. Neurons in the inferior olive show rhythmic oscillations, and this rhythmic activity may provide timing signals to the cerebellum. The inferior olive also appears involved in detecting errors and timing mismatches, providing teaching signals that drive cerebellar learning. The medulla also contains the pyramids, bundles of nerve fibers visible on its interior surface. These are the corticospinal tracts, motor pathways descending from the motor cortex to the spinal cord. At the bottom of the medulla, most of these fibers cross to the opposite side in the pyramidal decussation, which is why the right motor cortex controls the left side of the body and vice versa. Just lateral to the pyramids are the inferior olivary nuclei, which project to the cerebellum and are involved in motor learning and timing. Throughout the brainstem run ascending sensory pathways, carrying information from the body up to the brain, and descending motor pathways carrying commands from the brain down to the spinal cord. The spinothalamic tract, carrying pain and temperature information, runs through the lateral portions of the brainstem. The dorsal column medial lemniscal pathway, carrying fine touch and proprioceptive information, synapses in the medulla, and then crosses to the opposite side before ascending to the thalamus. Damage to these pathways produces sensory deficits, often affecting one side of the body depending on where along the pathway the damage occurs. Brainstem strokes, while less common than cortical strokes, are often more severe because of the concentration of vital functions in such a small space. A stroke in the medulla can affect breathing and blood pressure control, requiring immediate medical intervention to prevent death. A stroke affecting the cranial nerve nuclei can produce crossed syndromes, where damage to cranial nerve nuclei affects the same side of the face, but damage to ascending or descending tracts affects the opposite side of the body, creating a distinctive pattern that helps localize the lesion. The brainstem is also vulnerable to herniation, a condition where increased pressure in one part of the brain forces brain tissue through openings in the skull or between brain compartments. Transtentorial herniation, where the brainstem is pushed downward through the tentorial opening, can compress the midbrain and upper pons, producing rapidly progressive coma and death if not treated immediately. Tonsillar herniation, where the cerebellar tonsils are pushed downward through the foramen magnum, compresses the medulla, affecting respiratory and cardiovascular centers with similarly catastrophic consequences. The brainstem demonstrates that consciousness and higher cognition, while certainly dependent on the cerebral cortex, are not sufficient for life. The brainstem provides the foundation, the platform of basic arousal and autonomic regulation, upon which cortical function depends. You can have a functioning brainstem without an intact cortex, producing states like persistent vegetative state where arousal and sleep-wake cycles continue, but awareness and meaningful conscious experience are absent. But you cannot have cortical function without a functioning brainstem. The brainstem is, quite literally, the foundation of life. Part 4. The Limbic System. The emotional core. Emotion is not the opposite of reason. It's not a primitive remnant we'd be better off without. Emotion is essential for rational decision-making, for learning, for memory, for social interaction, for virtually everything that makes us human. And emotion arises largely from a collection of structures deep within the brain, collectively known as the limbic system. The term limbic comes from the Latin limbus, meaning border or edge, because these structures form a ring around the brainstem, bordering the division between the evolutionarily older brainstem and the newer cerebral cortex. The concept of a limbic system has evolved considerably since it was first proposed in the 19th century. Early anatomists focused on structures that appeared to be involved in olfaction, the sense of smell. Later, Paul MacLean proposed the limbic system as the emotional brain, distinct from the rational cortex. While we now know the division isn't that clean, the limbic system concept remains useful for understanding how emotion and memory are generated and regulated. The limbic system isn't a discrete anatomical structure, but rather a functional network of interconnected brain regions. Different sources list different structures as part of the limbic system, but key components typically include the amygdala, hippocampus, hypothalamus, cingulate gyrus, and several other structures. These regions work together to process emotional information, form memories, regulate motivation, and link emotion with perception and action. Let's start with the amygdala, which is perhaps the limbic system's most famous component. The amygdala is an almond-shaped structure, one on each side of the brain, located deep within the temporal lobe just in front of the hippocampus. The name amygdala comes from the Greek word for almond. The amygdala isn't actually a single structure, but a collection of nuclei, roughly 13 distinct clusters of neurons, each with somewhat different connections and functions. These nuclei can be grouped into several major divisions. The basolateral complex, including the lateral basal and accessory basal nuclei, receives sensory input and is involved in learning emotional associations. The centromedial group, including the central and medial nuclei, projects to brainstem and hypothalamus and controls emotional responses and behaviours. The cortical nuclei are involved in olfactory processing. This internal complexity allows the amygdala to perform multiple functions simultaneously. The amygdala is often called the brain's fear centre, and for good reason. It plays a crucial role in processing fear and other negative emotions, in detecting threats, and in triggering appropriate responses. When you encounter something dangerous, your amygdala activates before you're consciously aware of the threat, initiating the cascade of physiological and behavioural changes known as the fight or flight response. The amygdala receives sensory information from the thalamus and cortex, allowing it to evaluate stimuli for potential threat. This evaluation happens via two pathways that neuroscientist Joseph Ledoux termed the low road and high road. The low road is a fast, crude pathway directly from the sensory thalamus to the amygdala. This pathway sacrifices accuracy for speed, allowing rapid response to potential threats. The high road is a slower, more detailed pathway through the sensory cortex to the amygdala. This provides more complete information but takes longer. The fast pathway explains phenomena like jumping at a stick that looks like a snake before you consciously register that it's not dangerous. The amygdala responded to the crude visual information suggesting danger, triggering a defensive response before cortical processing could provide accurate identification. Better safe than sorry from an evolutionary perspective. A false alarm costs little, but failing to respond to a real threat could be fatal. When the amygdala detects a threat, it activates the hypothalamus, triggering the sympathetic nervous system. Your heart rate increases within a fraction of a second. Your blood pressure rises. Your pupils dilate, allowing more light in for better vision. Stress hormones like adrenaline flood your bloodstream, mobilizing energy stores and enhancing alertness. Blood flow shifts from your digestive system to your muscles, preparing you for action. You're prepared to fight or flee, all without conscious thought or decision. This response can be triggered not just by actual threats, but by learned associations, which is why someone who was in a car accident might feel fear when approaching an intersection, even when objectively safe. The amygdala learned that intersections predict danger, and it responds to that learned association automatically. This learning is remarkably robust. Fear conditioning, where a neutral stimulus is paired with something aversive, produces learning that can persist for years and is difficult to extinguish. The neural mechanisms of fear conditioning have been extensively studied. When a neutral stimulus like a tone is paired with an aversive stimulus like a shock, neurons in the lateral nucleus of the amygdala that respond to the tone begin to also respond to signals about the shock. Through synaptic plasticity mechanisms, particularly long-term potentiation, the connection strengthens. Eventually, the tone alone activates these neurons, triggering fear responses even without the shock. This learning happens rapidly, sometimes after a single pairing, and the memories formed are remarkably persistent. But the amygdala does more than just process fear. It's involved in processing many emotions, particularly those with high arousal or significance. It responds to happy faces as well as fearful ones, to rewards as well as threats, to anything emotionally salient. The amygdala appears to act as a relevance detector, marking stimuli as important and worthy of attention and enhanced processing. When the amygdala activates, it signals the rest of the brain that something significant is happening, something that deserves resources and attention. The amygdala has extensive connections throughout the brain, allowing it to influence multiple systems. It projects to the hypothalamus and brainstem, controlling physiological and behavioral responses to emotion. It projects to sensory cortices, enhancing processing of emotionally significant stimuli. It projects to the hippocampus, modulating memory formation. It projects to the prefrontal cortex, influencing decision-making. It receives input from all these areas as well, creating reciprocal circuits that allow complex interactions between emotion and other cognitive processes. The amygdala also modulates memory formation through its connections with the hippocampus. Events associated with strong emotion are remembered more vividly and more durably than emotionally neutral events. This emotional enhancement of memory makes evolutionary sense. Remembering dangerous situations, rewarding opportunities, and emotionally significant social interactions is more important than remembering mundane daily details. The amygdala, responding to emotional significance, signals the hippocampus to strengthen the encoding of the memory, ensuring that emotionally important experiences are retained. This explains why you probably remember exactly where you were during significant emotional events in your life, but can't remember what you had for lunch last Tuesday. The emotional significance of major events triggered amygdala activation, which enhanced hippocampal encoding, creating strong, durable memories. Routine lunches don't trigger strong amygdala responses, so they're encoded weakly if at all. Flashbulb memories, vivid recollections of surprising and consequential events, illustrate this phenomenon. People often report remarkably detailed memories of where they were and what they were doing when they heard about major news events. The surprise and emotional significance triggered strong amygdala activation, enhancing encoding. Interestingly, while these memories feel particularly vivid and accurate, research shows they're actually not more accurate than other memories, just more confident and detailed. The amygdala enhances encoding but doesn't guarantee accuracy. Damage to the amygdala produces striking changes in emotional processing. The famous patient SM, whose real name was revealed as SM after her death, had bilateral amygdala damage due to Urbach-Wiener disease, a rare genetic condition that causes calcium deposits that destroyed her amygdalae. She showed a complete absence of fear in situations that would terrify most people. She approached snakes and spiders without hesitation, watched horror movies without distress, and failed to recognize fear in others' facial expressions. SM understood fear cognitively and could describe it, could explain what causes fear and why people might be afraid in particular situations. But she didn't experience it herself. Her case demonstrated that the subjective experience of fear, the feeling of being afraid, depends on intact amygdala function. Interestingly, SM could still experience anxiety induced by carbon dioxide inhalation, which produces a sense of suffocation. This suggested that the amygdala isn't necessary for all forms of fear, but specifically for fear learned through external threats. SM also showed reduced ability to recognize fear and anger in others' facial expressions, though she could recognize other emotions normally. This demonstrated that the amygdala isn't just involved in experiencing emotion, but also in perceiving emotion in others. The amygdala may contain mirror-like mechanisms that simulate others' emotional states, helping us understand what others are feeling. Without an intact amygdala, this emotional empathy is impaired. Conversely, hyperactive amygdalae are associated with anxiety disorders. People with post-traumatic stress disorder show exaggerated amygdala responses to potential threats, even to stimuli that are objectively neutral. A car backfiring might trigger a full panic response in someone with PTSD related to combat exposure. The amygdala has become hyper-responsive, detecting threats where none exist, generalizing from the traumatic experience to similar but safe situations. People with social anxiety disorder show increased amygdala activation in response to social stimuli, like faces, particularly faces showing negative emotions or social judgment. The amygdala treats social situations as threatening, triggering anxiety responses that interfere with normal social interaction. People with generalized anxiety disorder show elevated baseline amygdala activity and heightened responses to uncertain or ambiguous situations. The amygdala has become overactive, constantly signaling potential threats. Various anxiety treatments, whether pharmacological or psychological, often work in part by modulating amygdala activity. Benzodiazepines reduce amygdala activation, decreasing anxiety in the short term though, with risks of dependence. Selective serotonin reuptake inhibitors, commonly used for anxiety and depression, appear to reduce amygdala hyperactivity over weeks of treatment. Cognitive behavioral therapy, which helps people reinterpret threatening situations as safe, appears to strengthen prefrontal cortex control over the amygdala, allowing top-down regulation of emotional responses. Extinction training, where feared stimuli are repeatedly presented without the aversive outcome, is a key component of exposure therapy for anxiety disorders. This doesn't erase the original fear learning in the amygdala, but creates new learning, an inhibitory memory that competes with the fear memory. The prefrontal cortex, particularly the ventromedial region, is crucial for this extinction learning. Strengthening prefrontal amygdala connections helps maintain extinction, preventing return of fear. When these connections are weak, fear may return after extinction, explaining why anxiety can recur even after successful treatment. The amygdala also plays roles beyond fear and anxiety. It's involved in processing rewards and positive emotions, It responds to attractive faces, to humour, to pleasant tastes. It appears to process emotional salience generally, whether positive or negative. The amygdala helps direct attention and resources toward emotionally significant stimuli, whether those stimuli signal danger, opportunity, or social significance. The amygdala's role in social cognition is particularly interesting. It responds strongly to faces, especially to emotionally expressive faces. It helps you read others' emotions, infer their intentions, navigate the complex social landscape. People with amygdala damage often have difficulty in social situations, not because they lack social knowledge, but because they don't spontaneously pick up on social and emotional cues that guide normal interaction. The amygdala is also involved in moral judgment. It responds more strongly to moral violations than to non-moral negative events. It appears to generate the emotional reactions, the feelings of disgust or outrage, that accompany moral judgments. People with amygdala damage show reduced emotional responses to moral violations, though their cognitive understanding of right and wrong remains intact. This suggests that moral judgment isn't purely rational, but involves emotional processing that the amygdala provides. Adjacent to the amygdala lies the hippocampus, a seahorse-shaped structure that's absolutely essential for forming new memories. The hippocampus doesn't store memories permanently, but it's required to transfer information from short-term to long-term storage, a process called memory consolidation. The hippocampus is actually a complex structure with several subregions: the dentate gyrus, areas CA3 and CA1, and the subiculum. These regions form a circuit, with information flowing from the dentate gyrus through CA3 to CA1, and then to the subiculum, which projects to other brain areas. This circuit performs a remarkable computational feat, taking complex multimodal experiences and encoding them in a form that can be stored and later retrieved. The hippocampus is particularly important for episodic memory, memories of specific events from your personal history. When you remember your first day of school, or your last birthday party, or what happened yesterday morning, you're retrieving an episodic memory that was encoded by your hippocampus. These memories include not just the facts of what happened, but the context, where you were, when it occurred, how you felt, sensory details like sights and sounds and smells. The hippocampus binds all these elements together into a coherent memory trace. The hippocampus is also crucial for spatial memory and navigation. The discovery that won John O'Keefe the Nobel Prize in 2014 showed that the hippocampus contains place cells, neurons that fire specifically when an animal is in a particular location. As you move through an environment, different place cells activate, creating a neural map of space. The hippocampus uses these maps for navigation, for remembering where you've been, for planning routes to where you want to go. Interestingly, London taxi drivers, who must memorize thousands of streets and routes, show enlarged hippocampi compared to control subjects, and the size increase correlates with years of experience. This demonstrates the hippocampus's plasticity, its ability to change structurally in response to demands for spatial memory. The hippocampus is vulnerable to damage from various sources. It's highly sensitive to oxygen deprivation, making it one of the first brain regions affected by conditions like cardiac arrest. It's also affected early in Alzheimer's disease, which is why memory problems are typically the first symptom. Chronic stress and elevated cortisol levels can damage the hippocampus, potentially contributing to the memory problems associated with depression and post-traumatic stress disorder. The hypothalamus, despite being tiny, weighing only about 4 grams, exerts enormous influence over your body's internal state. Sitting below the thalamus, to which it's intimately connected, the hypothalamus is the main interface between the nervous system and the endocrine system, between neural and hormonal control. The hypothalamus regulates fundamental drives and homeostatic processes. It controls hunger and thirst, monitoring blood glucose, osmolality, and various hormones to determine when you need to eat or drink. It regulates body temperature, triggering sweating when you're too hot, shivering when you're too cold. It controls circadian rhythms, your internal biological clock, through connections with the suprachiasmatic nucleus, which receives input from the eyes about light levels and uses that information to keep your daily rhythms synchronized with the external light-dark cycle. The hypothalamus controls the pituitary gland, often called the master gland because it secretes hormones that control other endocrine glands. The hypothalamus produces releasing and inhibiting hormones that travel to the anterior pituitary, triggering or suppressing the release of pituitary hormones that then act on the thyroid, adrenal glands, and gonads. This hypothalamic pituitary endocrine axis controls growth, metabolism, stress responses, and reproduction. The hypothalamus is also directly involved in the stress response. When the amygdala signals threat, the hypothalamus activates the sympathetic nervous system through its connections with autonomic control centers in the brainstem. It also initiates the hypothalamic pituitary adrenal axis, triggering the release of cortisol from the adrenal glands. Cortisol mobilizes energy stores, enhances cardiovascular function, and suppresses non-essential functions like digestion and immune responses, preparing the body for action. Different hypothalamic nuclei have specialized functions. The paraventricular nucleus produces corticotropin-releasing hormone, initiating the stress response. The supraoptic nucleus produces vasopressin, which regulates water retention. The ventromedial nucleus is involved in satiety, feeling of being full after eating. The lateral hypothalamus is involved in hunger. Early research suggested these were simple centers, on-off switches for eating behavior, but we now know they're part of complex circuits integrating multiple signals about energy balance. The cingulate cortex, a band of cortex wrapping around the corpus callosum, is considered part of the limbic system despite being cortical rather than subcortical. The ungulate divides into anterior and posterior regions with distinct functions. The anterior cingulate cortex is involved in cognitive control, in detecting conflicts between competing responses, in monitoring errors, in regulating emotional responses. It's active when you need to override a habitual response, when you're trying to resist a temptation, when you're dealing with uncertainty or making difficult decisions. The anterior cingulate appears to signal when increased cognitive control is needed, recruiting prefrontal cortex resources to resolve conflicts or correct errors. The anterior cingulate is also involved in pain processing, not just the sensory dimension of pain, but the affective dimension, the unpleasantness. Damage to the anterior cingulate can produce painous embolia, a condition where people can detect painful stimuli but don't find them bothersome. They can tell you exactly where it hurts and how intense the sensation is, but they're not motivated to escape or avoid it because the emotional component is absent. The posterior cingulate cortex is involved in memory retrieval, in spatial orientation, in maintaining a sense of self. It's part of the brain's default mode network, regions that are active when you're not focused on external tasks, when you're mind-wandering or remembering the past or imagining the future. The posterior cingulate shows altered activity in various psychiatric and neurological conditions, including depression, Alzheimer's disease, and schizophrenia. Several other structures are often included in the limbic system. The mammillary bodies, small rounded structures on the underside of the hypothalamus, are involved in episodic memory and show damage in Korsakoff's syndrome, a memory disorder associated with chronic alcoholism. The septal nuclei, located below the rostrum of the corpus callosum, are involved in reward and reinforcement. The nucleus accumbens, part of the ventral striatum, is crucial for motivation, reward processing, and addiction. The fornix, a bundle of nerve fibers, is the main output pathway from the hippocampus, carrying information to the mammillary bodies and other structures. The limbic system demonstrates that emotion and cognition are not separate processes but deeply intertwined. Damage to limbic structures can produce not just emotional changes but cognitive impairments. Conversely, damage to cortical regions can produce emotional changes because those regions are part of circuits that include limbic structures. The prefrontal cortex, for instance, sends massive projections to the amygdala, providing top-down regulation of emotional responses. Depression and anxiety involve not just limbic dysfunction but altered connectivity between limbic and cortical regions. Understanding the limbic system helps explain various psychological and psychiatric phenomena. Why do emotionally arousing events produce stronger memories? Because the amygdala modulates hippocampal encoding. Why does chronic stress impair memory? Because elevated cortisol damages the hippocampus. Why do people with anxiety disorders perceive threats where others see neutral stimuli? Because their amygdalae are hyperresponsive. Why do decisions often feel influenced by gut feelings? Because limbic structures, processing emotional information, influence prefrontal decision-making systems. The limbic system is also central to psychiatric disorders. Depression involves altered function in the anterior cingulate, amygdala, and hippocampus, along with abnormal regulation by the prefrontal cortex. Anxiety disorders show hyperactive amygdalae and altered fear extinction in connections between the amygdala and prefrontal cortex. Addiction involves dysregulation of the nucleus accumbens and its dopaminergic inputs, creating powerful associations between drug cues and reward. Post-traumatic stress disorder involves enhanced fear conditioning in the amygdala and impaired extinction, along with altered hippocampal function affecting memory for the traumatic event. Treatments for these conditions often target the limbic system, either directly or indirectly. Antidepressants affect neurotransmitter systems throughout the brain, including in limbic structures. Anxiolytics dampen amygdala activity. Cognitive behavioral therapy works in part by strengthening prefrontal control over limbic responses, teaching people to reappraise situations and regulate emotions. Even meditation and mindfulness practices show effects on limbic structures, particularly the amygdala, potentially explaining their benefits for stress and anxiety. Conclusion: the brain as a unified system. We've journeyed through the brain's major regions, from the cerebrum's cognitive complexity to the cerebellum's precision timing, from the brainstem's vital functions to the limbic system's emotional processing. But understanding the brain isn't just about cataloguing structures and their functions. It's about appreciating how these regions work together as a unified system. Information flows continuously between regions. The sensory cortex sends information to the parietal and temporal lobes for processing. Those regions project to the prefrontal cortex for decision-making. The prefrontal cortex sends plans to the motor cortex for execution. The motor cortex sends signals to the cerebellum for coordination and the basal ganglia for action selection. The brainstem maintains arousal and autonomic functions that allow all this to happen. The limbic system evaluates significance and modulates memory and emotion. These are not separate systems, but interconnected networks. A single thought, a single action, a single moment of consciousness involves activity across multiple brain regions simultaneously. The networks that underlie even simple tasks are staggeringly complex, and we're only beginning to understand how they function. The brain is also remarkably plastic, capable of reorganization throughout life. Learning strengthens synaptic connections between frequently coactive neurons. Damage to one region can sometimes be partially compensated by reorganization in other regions. Experience shapes brain structure, from London taxi drivers' enlarged hippocampi to musicians' enhanced auditory cortices to the changes in prefrontal limbic connectivity that occur during adolescence. We've learned an enormous amount about the brain in recent decades. Neuroimaging techniques let us watch the living brain at work. Molecular and genetic methods reveal the mechanisms underlying neuronal function and plasticity. Computational models help us understand how networks of neurons give rise to complex behaviors. But vast mysteries remain. How does neural activity give rise to conscious experience? How do billions of neurons, each following simple electrochemical rules, produce the rich inner life of thoughts, feelings, sensations, and experiences that characterize human consciousness? We can map which brain regions activate during different mental states, but mapping correlation is not explanation. The relationship between brain and mind, between neurons and consciousness, remains one of science's deepest mysteries. We don't fully understand many brain disorders. Why does schizophrenia emerge in early adulthood? What causes autism spectrum disorders? Why are some people vulnerable to depression while others are resilient? How does consciousness emerge from a coma, or fail to emerge in persistent vegetative state? These questions remain partially or completely unanswered, but every year brings new discoveries, new techniques, new insights. We're developing more sophisticated imaging methods, genetic tools that allow manipulation of specific neurons, computational models that capture more realistic brain dynamics, therapies that target specific circuits rather than flooding the brain with drugs. The brain is the most complex structure we know of, but it's also the tool we use to study itself. The human brain investigating the human brain is perhaps the universe's grandest recursive loop, and every discovery, every insight, every question answered raises new questions, deeper mysteries, further horizons to explore. The 3 pounds of tissue in your skull right now orchestrated your reading of this script, processed every word, integrated the information with what you already knew, generated understanding and perhaps wonder. That same tissue is what allows you to love and fear and hope and dream, to remember the past and plan the future, to to create art and solve equations and contemplate your own existence. You are your brain in a very real sense. Your personality, your memories, your thoughts and feelings, and the continuity of your selfhood across time all emerge from neural activity. That's not a reductive claim that diminishes human experience, but a recognition of how remarkable the brain truly is. That neural tissue can generate consciousness, that electrochemical signals can produce the feeling of being someone, experiencing a world, is perhaps the most astonishing fact in all of nature. And with that, we've reached the end of tonight's journey. If you genuinely enjoyed this deep dive into the architecture of human consciousness, do me a favour and hit that like button. Subscribe if you want more explorations like this. And drop a comment letting me know what aspects of the brain fascinated you most, what questions you still have, what topics you'd like me to cover next. The brain is endlessly fascinating, and we've barely scratched the surface, but that's the beauty of it. There's always more to learn, more to discover, more mysteries waiting to be unravelled. Until next time, take care of that £3 universe in your skull. It's the most valuable thing you own.